Hello and welcome to this IBTV Spotlight on starting materials, where we're asking questions on quality. I'm joined by Doug, Kevin and Carlos here to discuss a little bit about how we can ensure quality into the different stages of the starting materials process in feeding the CGT industry. So just to start with, we're all from different backgrounds and perspectives. We're looking at this from different lenses. For you and your experiences and in your worlds, what are the biggest challenges and bottlenecks and just biggest issues and bugbears really that are going on with starting materials from your point of views?
Let me start. Sure. I, we're mainly an upstream provider of the source material. Knowing specifically what a client wants, and a lot of them are anticipating because they may not, historically, not every collection site had the ability to have flow cytometry, for example, right? They may say, hey, since you don't have flow, just get an MNC dose, right? But to us, since we actually have that facility near us, knowing exactly what the cell is you need and the minimal, the minimal acceptable amount really helps us optimize that because we're able to do obviously a peripheral blood starting, but we're also do a mid count of the product and then use that to estimate to get to the end. Because obviously, if you over collect the donor, if you collect way more than you need, and the donor has to sit there for a long period of time, then they're less likely to want to come back, right? Or if you do everything that the MNC, for example, mononuclear cell, they said, oh, if you collect that, then you're going to collect enough T cells or NK cells or whatever, and then you wind up going, well, no, not really. We were just estimating. So for us, the biggest barrier to that is just the lack of precision of what they actually need, of what's actually needed. I think the second one is the fact that everybody, every vendor, every group we talk to has a different way of telling us. So our nurses don't have to be experts in one thing in 50 things, and they have to make sure, oh, it's this group, so we have to do it this way. And we're just worried at scale that we're going to make a mistake because it still takes a human being to sort of go, oh yeah, it's this.
And Terumo is a global technology provider. We provide technology, services, and solutions for all the way from starting material, blood component separation, cell therapies, and also for plasma collection. So when we look at these procedures and these collections, some of those collections are very complicated, right? Especially if you're collecting from a sickled patient, right? Sickle cell disease patient. Those are hard collections, especially looking for 34, CD34 cells. So what we provide is we provide technologies that enable all the cell collectors to collect those. In addition to that, we understand that different disease, as I was mentioning, are different patients are different. So we also look at optimization services, how if it's a particular complicated collection or you want to collect, how do you optimize those collections? So this is where we contribute to enable the collectors and how can we help them, like providing the best devices and best machines for sure, but also providing those services which can help optimize those collection. And talking to physicians, talking to cell collectors, understanding what their pain points are, what their needs are. We also provide education services. Like not everybody in the biotech, like I know you're very well aware of apheresis, you're very well aware of it, but like there are people in this ecosystem who do not appreciate the apheresis enough. Right. So providing that education, what is apheresis? What are the key variables? What are we doing with this? Like how can you optimize? What can you do? What to look out for when you're doing your SOPs? So those kind of education is also provided. Like for example, me and Erin, we contributed to a chapter at ARM when they were putting the ACell together, the chapter on starting material considerations, for people to appreciate what all variables go into it and how to think about it. So that's where we come in from our lens perspective.
I think education is definitely important, right? So OrganaBio is a cell supplier as well. We focus on peripheral blood material, but also cord blood material. Part of the challenge that we have is educating the individuals that we work with on the various options that are available for collecting apheresis material, right? Even on the Terumo devices, you have different collection parameters that you can use, and there's not at least one consistent collection parameter across individuals. So oftentimes, one supplier might be collecting a certain way, another supplier might be collecting the other way, and when you try and compare them at the end of the day, there's slight differences. Not only because of collection parameters, but I think the other challenge is the donor differences. And the fact that we are dealing with live donors, there's challenges in collecting material from live individuals who, have their own sets of challenges, right? Their own scheduling issues coming in. But I think that's been one of the main challenges, right? Is how do you maintain an active donor pool? How do you maintain donors that are healthy enough, at least on the healthy side, to provide high quality collections? How do you do it in a safe way where there's enough time from their previous collections to allow their cells to reconstitute? And so I think there's a lot of challenges that you have to deal with when you're dealing with donors, when you're dealing very specialty collections, because you want the material that you're collecting on the front end to be very high quality. So when it goes in through the rest of the processes, you have that high quality material throughout the procedure. Yeah.
Yeah, since you're talking about education, I know we all agree the education is not just the donor, right? It's also the staff, it's also the nursing, it's everyone who touches us to know what's important, what parameters can we adjust. Vitalant, 28 states, we take care of a lot of patients. We have blood donor side. We also have it on the cell therapy and the cell starting side, and it takes a while for the nurses to educate the staff on the input parameters and what this means versus that means. And you want staff that are hungry for that information, because you can't predict everything that's going to happen, and educate them all up front. And quite honestly, at least me with my ADD, I wouldn't have picked it all up the first time you told me anyway. So having that ability that people can call and reach out and say, hey, this doesn't look right. That's the beginning, I think, in a lot of times of resolving instances where you may not have gotten a good collection, but because they saw something that they went, oh, I need to escalate this to the technical person or the doctor or another nurse, we're able to fix that. So I think at the end of the day, knowing a lot about donors and educating them, incredibly important, especially if those donors are patients, but also the staff, to make sure that they understand that this is not, this is a GMP, this is a GTP environment. And so there's certain things you have to do to make sure the quality in is what happens with the quality out.
Yeah, another thing I've heard from starting material, and maybe it continues like you mentioned, I've heard from people who are collecting in the biotechs is like there's a communication gap. What I mean by that is, collectors have told like, oh, we don't get the feedback. Was it a good collection or not? We have provided cells. And they say like, oh, collect MNCs, but they are targeting NK cells.
Correct.
Or CD3 or subpopulation of CD3. If they can tell that, maybe it's helpful for us to know, is it whether they have capability to do analysis or not? It's different between sites. But it will be helpful for them to know, are we collecting good? Should we continue the path that, is it a good collection or not? So I've heard that. I don't know how much it has improved over the years, but that was like a year ago. That was the discussion. I still hear that SOPs are designed with, oh, collect on MNC, while the target is CD3 or NK or some specific subpopulation with an MNC.
Well, I think the way Carlos was saying up front is spot on, that if you go in to collect, you can collect based on the length of time the person's on the machine. You can collect by the number of total blood volumes of a donor processed, right? You can collect based off of MNC. You can go based off of also the specific subcell type, and each one of those has its own plus and minuses. And there's a lot of the industries beginning to understand that if we just say what the minimum sample product needs to be, and then allow that team to sort of use potentially any of those mechanisms to get to that. If we could do that, to say, whether you have it for 2.5 total blood volumes or 4 hours, or you go for a specific dose, as long as the cells have so much viability, there's a minimum number of this specific cell type, it'll be okay. So I think that was early on. Being overly prescriptive was a way to hard code a quality product. But if you're already using people that are accredited by ABB, FACT, whatever, you already have a quality system that ensures a quality product coming out. And I'd love to see where we could leverage those credentials that a lot of us already have to say, hey, can we collect it however we know allows us to collect it correctly, and just tell us what the final bag, kind of like on the red cell side. I know what the minimum red cell dose or the minimum platelet dose. Just give me that, and then I can design a system around that.
Oh, and I think that's a good point too. I think to better inform that, we need to have a very tight eye and good quality control, right? What are you measuring after the collection? When are you taking those readouts? Is it for cryomaterial? Is it pre-cryopreservation? Is it post-cryopreservation? How soon after are you processing that leukopak in an apheresis sense, right? From the time of collection to when it goes to your processing site for freezing or for downstream isolation of isolated cells. And I think having good quality control, taking those viability readouts, taking the immunophenotyping at stages where it's most critical, right? Pre-freeze and post-thaw, you can really have a good sense of how good was the collection, how high quality are the cells, and then that can kind of inform kind of what you guys are doing at Terumo on the collection side. Can we modify things on the collection to kind of improve the downstream isolation or the downstream quality of the cells?
So yeah, we've raised ideas around the communication pathways and education as well, and we're looking very much at the kind of collections process. How is that kind of replicated, but looking at the other end, who's receiving the starting material and who's kind of talking about this from a kind of therapeutic development point of view? Is that same communication pathway happening there? And what is what kind of education process? How are people scrutinizing their starting materials, I suppose, to start with?
Yeah, I can start. So I think what I have heard from industry, especially because when it started, we are talking about standardization of collection, but the cell therapy started well before that, when that was not existing. So the teams in the labs have that thought process: whatever comes in, we can handle it. And as the scaling, we are reaching more patients, they understand that better cellular material makes their life easier, and they can make the end processes much better if they don't have to do a lot of upstream cleaning of the process. But if you talk to them, you will hear most of the therapy developers is like, yes, we can handle, because we have different kind of selection processes, we have downstream processes, which allow us to help the cells. Now, if you ask them, further interrogate and ask, like, will it be better to have a cleaner product? Then most of them will say yes. I would say all of them would say yes, but maybe most of them will say yes. And cleaning the product or having a better product, better collected product, what they're looking for, whether it's CD3, NK, or cells, that will be good if you talk to them. That's how they're looking at from a communication perspective. How do you get? Now, if you already have a regulatory approval on a process, they're always hesitant to change. Now, this is where the discussion needs to happen with the biotech providers, like what has changed in the process. And FDA is pretty open. If you make sure you're making the right product and you have the right controls, and go to them, it's like, we're making the process better, but the end product is still the same. You can take that discussion forward. That's how I think about it.
No, I think the process improvement aspect is very difficult, because it gets very hard-coded once the FDA has approved a single process. It's a big effort, right? I think from the lot to lot, collection to collection, definitely the communication has to include, if not immediate feedback, at least periodic KPI indicators to look for metrics, right? To ensure that the quality that you agreed to up front is maintained over time, right? Because you bring in new staff or whatever, or you're collecting on different days, and there's lots of variables that you didn't engineer for up front that occur over time. I think from a collector, I do think downstream, we don't hear of very many things unless it's just a large failure. Although the industry says, 15 to 25%, that's a little high, but about 15% of some of the manufacturing fails. Well, how much of that is things that collections could have helped with? And obviously, we don't know the answer to that. Some of those things are just beyond our control. But if you're dealing with a patient, and you're collecting a patient who's been heavily treated with chemo and radiation, is there a model, and we don't have one yet, but is there a model where for certain patients that have a high probability of moving on to a cell therapy, to collect them before they've had a lot of therapy and a lot of radiation, and their cells have been bombarded by these toxic drugs that might increase their success? We have to have a payer solution before we can get to those types of things. But I know our nurses are so, well, operationally us as well, but the nurses are so committed to providing the best product that oftentimes I feel like they don't get enough information that could help them do a better job the next time.
Yeah, I think for us, the communication has mostly been on the front end, right? Is we're delivering typically fresh material, sometimes cryopreserved material, and the first piece of communication that we want to get from the client or partner is, did you receive the material? Was it okay? For frozen material, was it cracked? Was something wrong with the bag? For fresh material, did it arrive on time? Did it arrive at the temperature that you expected it? Were you able to kind of do some initial quality testing to compare kind of the data that we provided on a C of A, knowing that, there's differences in how labs do measurements. They might not use the same exact equipment, but is it somewhat comparable? But oftentimes, then I think the communication tends to fall off a little bit because some individuals have longer processing times where they're working with material for 5, 6, 7, 8 weeks at a time, and you tend not to hear something for a couple of months. But we always try our best to kind of have that communication, see, hey, did that donor work? Might it be beneficial to test multiple donors to see if there was a failure, if it was due to donor variability or something else along the process? And so I think definitely crucial to have that communication with your partners to be able to see what's working, what's not working, how can we improve, and what can we do potentially better the next time. Yeah.
And what potentially, if we're thinking about starting materials and a question of quality, what's missing here in this? Is there tools and technologies that can't be brought in for certain things? Is it donor variability that makes this particularly hard? Like, is there a kind of a space? Is there anything missing that we're kind of not really addressing, or we're not able to adopt as we start looking forward and trying to make this, let's say, everybody's lives easier?
Yeah, I mean, I think for us it would be just having a well-characterized product. Provide as much characterization data as you can up front. If that's immunophenotyping, if that's HLA, if that's TNC, if that's viability, if that's donor demographics, right? Age, sex, BMI, smoking status, different IDM testing. The more information I think that you can provide ahead of time, the more valuable it becomes downstream when you're starting to compare data across multiple donors. You can start to see some similarities or some differences and say, okay, I'm starting to identify my ideal donor because I have all of this data, right? And not just on the donor side, right? Collection parameters. How was this, in our case, apheresis, how was this material collected? What device was it collected on? The more data I think that you can provide up front from both the collection parameter side, the donor side, the quality control side, I think the better. But the challenge is there's not one standard operating procedure across, all of these different types of, companies. So it makes it very difficult to have a one size fits all.
And I completely agree, because the sort of the point that you started off with, characterization of what sort of cell types, and I think we as quickly as we can, I'd like to see us move to where we can provide some of that data at the end of the characterization so the facility doesn't have to do all that when they receive it. However, we all know flow cytometry, blood is a non-homogeneous liquid, and flow cytometry, there's enough variability that it's really hard to compare labs against labs. There are some groups that do that. I'm really wondering, and this is just sort of spitballing, but is there a way to do like an international normalization ratio where labs that are a limited number of labs that are part of this process agree to figure out how my test run against your test? Because when I have to set up, and maybe some of my setup for my flow, I'm more conservative, and maybe you're a little less conservative, but if we know what those are and have a little fudge factor, but INR factor that says, oh, in general, Dr. Land at Vitalant, they run 85% and you run 115%, and you're kind of in the middle. And once we understand those ranges, then at least there's some consistency across us, because even if you use the ICH protocol, there's enough variability in there that's not a true apples to apples comparison. So I do think at some point, if we're moving that direction, we're gonna have to do some of the hard work in our own labs to redefine that. And if you're getting into post-thaw, most of the kits aren't really optimized for post-thaw, because the antibodies don't bind as well to the antigen, and those the cutoffs for the kits aren't really designed for that. So I think if we're going to move to that, which I agree is a good way to go, then the whole industry is going to have to sort of take a step back and reimagine how we can make our results more consistent across the labs.
I think that's all we have time for. So thank you, everybody, for joining me today. It was a really interesting conversation.
Thank you.